Yeah, just the talk I put together for Melton Botanical Society, and basically it just talks about everything that I've done. I've been looking at Pimelea now for about 14 years. Started my PhD in about 2008. So I'll just give you a rundown on everything I've learnt, where I think we should be going, the future, what I've learnt. And I mean, Megan features heavily too, who's on the call, I can see, who was my supervisor and is an amazing friend who supported me through all this. And yeah, it's been a great journey. I love every minute of it. And it's interesting, the photographers there, I actually love taking photography of grassland species, and it's not easy. Not easy to get a good shot of them. And that's one of my shots there at Banktree Grove. If you have questions, please ask. Let's hope we can get going. Okay, the genus Pimelea, there's about 150 worldwide, 110 are endemic to Australia. There's about, the rest of them are from New Zealand, Timor and New Guinea, so it's a Gondwanan species. One of the most common Pimeleas is Ferrugina, which is, I don't think I put that picture in there anymore, it's a pink Pimelea, but a lot of people see it's a Western Australian one. 36 of the 110 species that are endemic are, 36 are found in New Zealand. 18 of the Australian ones are rare or threatened. So it's not a plant that's easy to propagate and easy to get going. Here's a few more. That's Curviflora. Pimelea spinescens, subspecies spinescens. There's two subspecies, the spinescens and pubiflora. Spinescens is endemic to Victoria, and so is pubiflora. It has spinescent stems, which I'll show you a picture of hopefully. Yeah, there's some spiny, pointy stems. They're not spines as such, they're pointy stems, which are quite nasty when they're dry. And then we have pubiflora, which is the Pimelea Rice-flower, and I'll show you what that is in a minute. And predominantly its difference is it has hairy flowers, hairy sort of globule seeds, as you can see quite clearly there. It's also a much more yellow colour flower, and it's a little bit bigger, and I'll show you where it's found in relation to Pimelea spinescens. Interesting, pubiflora was thought to be extinct, basically, and in 2005 we found a population in Natimak area of about 250 plants, 2,500 plants, sorry, and then in 2007 we found another group of them in Minyip of about 115 plants, and since then we've found two more small populations in between those two areas, and we have been able to propagate and grow them as well, similar to what I can do with Pimelea spinescens. Subspecies spinescens, we can propagate them quite well, which is great considering how rare and threatened it is. There we go, there's a picture of pubiflora in its natural habitat. I think that's Natimak, that population is. The distribution, this is the sort of creamy areas is where it should be, the lighter cream, the darker creamy areas, sorry, that's the volcanic plains, as well as the black clay soils around Echuca, the northern plains, I suppose you call them. And the green areas is where it probably is found, and the red areas is where we definitely know it's found. They're all little dots, and you can see they quite clearly follow roads normally, and there's quite a large area on the outskirts of Melbourne. The main reason why those areas are really there is because it wasn't superphosphated. It was just left as areas of, you know, we weren't value adding to it, and we'll just leave it as it is. And so Pimeleas just flourished in those areas where they weren't destroyed, and unfortunately they're right in the middle of development areas now. But that is where the grassy plains, the Western Grass Plains National Park is supposed to be. Where it's covered by the EPBC Act, it's critically endangered federally, and the FFG, it is threatened, which is a state act. We have a recovery plan in 2006. There's an action statement in 2008, and the latest conservation advice was 2016. And currently we have been revising the plan over the last couple of years. We've got that back up again with the federal government, and it is currently out for public comment on the federal website. You're very welcome to go there and download it, have a read. It is quite an extensive document, and any advice or anything, any comments, any way you could improve it, because it is written in a way that we want land managers and stakeholders to be able to use it to get funding to do things with Pimelea. So if you think it could be augmented and improved, please do comment on it. Get the comments maybe to me, or you can do it yourself. You simply email, there's a website and an email, and you can simply email your comments into the federal government by the 11th of February. It's been available since last year. It's been available for 3 months. So it's pretty bad timing for us. But if you do get a chance, please do it. And I suppose most people will realise less than 1% of volcanic plain exists in its natural state now. It's critically endangered, the actual, the ecological, was it EVC? Yeah, vegetation community. So everywhere. Pimelea is a bit of a flagship species for this, this grassland, and we need to look after it in all its shapes and sizes. Okay, spinescens, back to that. It's a dioecious plant. It means that each individual plant is either male or female. There is some in-betweens. This is a male plant on this side, and you can quite clearly see the stamens, which are quite orange with the pollen on it. And over here, we have a female with her central stigma, and she does have immature stamens down the bottom there. So the plant was hermaphroditic, and it evolved to be dioecious, and we think that's an adaptation for the very, very hot climate that it exists in. And there are hermaphrodites that we find within populations. They're definitely still present there. So it's a subshrub, very small shrub. It definitely has woody stems. It's usually quite small, 5 to 30 centimetres height. It's either prostrate or upright. And there's another very classic presentation of it with its spiny, pointy stems. And that's a female there in her presentation. This is another great photo that I took that clearly shows the differences between males and females. To the right over here, we have a female. Now she's in full flower, and what she does is she holds her little stems down. She's hiding her flowers, saying, "Don't look at me, don't look at me." And the male over here is saying, "Look, I've got lots of pollen, come and get me, come and get me." So basically all of the pollinators go there and they all clamber over everything. The other thing is this plant is highly nutritious and palatable. So we think that in Gondwanan times, perhaps that the female didn't want to be eaten. So she wasn't seen as obviously maybe. It's a possibility, but they're definitely quite differently presented when you can see in the grassland. This is another great presentation of the plants in the middle of summer. When you've got the themina in its full glory, all flowering, putting out its little stems, you cannot find the Pimelea. You really need to look for Pimelea in winter. It's a winter flowering species. That's when you'll see it, and that's the best time to do your surveys. In summer, it's really hard to find. There's a Pimelea right in the middle there. There it is. There's a little Pimelea stems, a little bit of it's always green. It never is not green, and it's right in the middle there. You can't really see it. It's a bit of a bad photo, but it is there, and it's very difficult to find in summer. So don't go looking for them in summer, not unless there's been a fire through. You probably might find them then. So its seed, we think it might be named the common name, Spiny Rice-flower. It could be due to its small seed, like a little rice grain, as you can see here, and it's glabrous. There's no hairs on the seed of spinescens. It's insect pollinated, and as I said, it flowers over winter from April to August, so that's quite different to all the other species of plants that we know. A lot of grassland plants flower during autumn, flower over summer, spring and into summer. But we think Pimelea flowers in that time when nothing else is flowering. So it's supplying the pollinators of the time with a little bit of nectar. There's a tiny little bit of nectar or water at the bottom of every flower. And it also has a smell. It's from the Daphne family, the Pimelea family. So when you see a lot of them in flower, stick your nose in if you like, have a smell, and there is a beautiful perfume, and certainly in the seed orchard I've seen that. It's insect pollinated, and it has a little fruity drupe, which actually is quite fleshy, and I'll show you that in a minute. And we thought with my PhD, we weren't sure was there an issue with pollination with the pollinators. So I went out and had a lot of look at them and seen what was actually there, and there was lots and lots and lots of different species of flies. We've got 1,000 species of flies in Australia. It's very difficult. A lot of them aren't named, but that's what I basically found on them. Many different flies. It's not really a moth, and also it's sort of covered with animals. This photo here has caterpillars. There's a spider in the corner here. There's a spider over here. There's a little golden beetle here. It's covered with insects and animals. So I think of it a bit like a rainforest tree in a grassland. It's just very small, because in the middle of summer, it's still got green, and in through winter, and things are always eating it and using it and hiding in it and hunting in it. And it's basically, its own little biodiversity network within a plant. There was ants all crawling over, and they definitely did take seed away. And we definitely found them taken off, but not huge amounts of seed. So they take them away and put them somewhere. We have flies doing their things on it. We had some moths. That wasn't very normal, and that's actually on Pubiflora. That was actually a moth that was found on Pubiflora. And then we had the interesting aspect of the spiders hunting the flies. I couldn't understand why this fly wasn't moving, and when I really got in and looked at it, I saw that there was a green spider with a yellow abdomen basically eating it. And I saw quite a lot of versions of that particular spider. It's a close-up of the spider on the ground, and it looks like it has a Pimelea flower on its back to me, or its abdomen. There was white forms of this, and there was also dark forms of this particular spider too. So I saw this spider on lots of different populations across the state when I was doing my PhD, having a bit of a look at them. Now give you a bit of a talk about the roots. I had a permit to dig up these roots for scientific purposes as they were going to be destroyed anyway. It was the Christie's Road site. This plant is probably a couple of years old. It's got quite a distinctive taproot. We weren't even sure early on when I started my PhD if they were prostrate, if they grew, lignonis sort of across the ground, but definitely they have taproots, and they also have like a claudiform taproot. That's a much bigger specimen, and they definitely send down this massive taproot at the start, which could average up to 60 to 80 centimeters into the ground, depending on the hardness. And then they would send up these amazing laterals as well. They didn't seem to have a lot of laterals up top, and it depended on what else they basically came across. And also when we do translocations, you take, hopefully if you get the bottom of that taproot, we think that that will actually enable them to survive essentially. And so here's a bit of a landscape view of what we think is going on. There's the surface of the soil up here. Here's the taproot, that's an older plant. Down the bottom, the start of it, and the soil. And what happens is they get these underground stems over time. If you know anything about clay soil, it's like concrete in summer, and it's quite soft when it gets really moist. So what's happening over time is the plant is slowly sinking into the soil. It's sort of during the wetter times, it sort of can move around a little bit and sink down, but then in summer it's like concrete, it won't move much at all. So it's sort of a suction slowly. And I did wonder if the lower in the soil it was, the older the plant probably is, which is probably true, but it was difficult. And we did try to age them in my PhD, but it was hard to get, you had to get a root because as you would realise in a grassland, a lot of the biomass burns off on the top. So that biomass can be quite young and you need the oldest part of the plant. And when we sent off some of the specimens, they came back as being all 50 years old, even though the plants were quite big and different sizes. It didn't really give us a huge amount of information, which was disappointing. Few more roots to show you. Essentially, you might have thought that was a different plant over here, but it's not. It's all connected back to this one taproot, so it's definitely still part of that plant, even though it's got this really interesting underground stem soil that you wouldn't have known unless you dug it up. And this was the Christie Rose area when we dug it up, and we knew that plant was a big plant, so we went right around as the heart was, long way around to see if we can get every single root out of it. And then when we got it up, it was like a archaeological dig. We were able to extract it from that area. And sure enough, it had extended about 2 meters one way and about 1.5 meters another way. So it was a very extensive area that this plant had occupied underneath the soil with its laterals. But in saying that, plants actually quite happy to be next to each other. The reason why that particular, that's a bit abnormal is there was a massive rock there when we dug it up. So the taproot had got to that point and then sent down 2 taproots, which is a bit unusual, but obviously it had been there a very long time and doing just fine. It had this 2 sort of taproots, and so it's very adapted to its soil type and its area where it's living. This was, we thought, one plant when we saw it. We thought, oh, just a cute little one plant sitting there. But if you look a bit closer, you can see there's 3. And when we actually dug them all up, that's all we got. So these plants are very happy to be right next to each other. They can germinate all together and they won't necessarily knock each other off. They'll quite happily, I mean, as time went on, maybe only one of those plants would live, but certainly at this stage, they're probably easily 2 or 3 years old at this stage. They were still quite happily right next to each other in the soil. So what were the major issues for Pimelea spinescens? This is what I looked at in my PhD. The reason for its listing as it being endangered is it's undergone a very large decrease in the numbers and the size of populations generally. It's severely fragmented. So all the little roadsides aren't connected anymore. All the little railway routes aren't connected anymore. Only 5% of the known populations are protected in reserves, with many populations vulnerable to roads, railworks, burials, industrial, agricultural areas. We didn't really know, there was rare recruitment reported. We didn't really know any recruitment that was definitely going on, and no one really talked about whether there was new plants within populations. There's a general lack of knowledge. How long do they live? What is their natural recruitment? We didn't even know when they germinated when I started. And as I said, 5% protected in reserves and lots of threats with road, industrial, and lack of management generally. People weren't managing consistently. Some areas don't burn at all, some areas did. So my PhD I wanted to identify what factors of the biology, ecology, and management significantly affects this species in situ recruitment potential. So we got a hold of all the populations that we knew of, and I selected 16 across the whole state. There was ones north, I'll show you a picture in a minute, and we set a site area, we looked at the population, we looked at sex ratios, we set up some permanent quadrats, individuals are marked, and we collected seed from about 3 to 7 females. There we go. Okay, there we go. There's my study area. That's what I looked at, where the plants found, which we had a look at before. And these are the sites. So we selected quite a few sites, 4 sites around Melbourne. We had 4 sites up in the more the goldfields plains up there and the edge of the plateau up here, and then a lot of sites across the west. We tried to select sites that had larger populations, medium-sized populations, and small populations to try and capture a broad reflection of what is actually out there. And also Ararat was a bit of an outlier out here, but we did it sequentially. We just looked at all the populations, grouped them, and just picked the middle ones in the middle, so it wasn't picked as such. So measures of reproduction to evaluate a population's recruitment potential. I needed measures. Most grassland species reproduce by seed, so the annual seed quality and quantity is quite important, as most are not considered to produce long-lived seed banks. So the logical measure was the plant's ability to produce seed, and it's also corresponding viability and eventual germinability. So the seed percentage was divided by the seed batches. Oh, that's right. I collected lots of seed. There's my little seed bags, and we have a protocol for this. We've put little stockings were the best method, with little like thread ties on them, I suppose. And I reuse these over and over again to collect seed. You need to put them on sort of in July, and you need to probably take them off when it starts to get warm. I usually do it around the end of October, early November, because the seed will naturally fall off the plant then. With viability, I looked at, it's an oily seed. I'm not sure if anyone's Greek here, but Pimelea or Pimele means soft, fat and lard. So this seed is an oily seed, it floats, and it also contains oil if it's a good viable seed. So the easiest way to actually look at viability was to squash seeds. And it's also a really effective thing to do in the field. If you don't know if the seed you're collecting is very good, is to squash it in a paper bag, which you have handy, and see if it's actually producing some oil. Is it producing good seed? Unfortunately, it's the easiest way to do it. There is other methods which are very laborious and a lot more expensive, a lot more time consuming, and actually don't give you the best results. Germination, we had looked at germination. So we collected from the seed I collected from the random plants across all the state on different populations. There was 3 to 7 at each population. We collected them and we had to do germination tests, which I'll show you in a minute. There was 35 different treatments that I put them all through. It was a very long and drawn out process. I won't go into it all. And then we still looked at germinates in the field. So whether I saw them or not, and especially I started my PhD in 2008 and 2009, I did all the sites and I noted whether there was germinates, and we went back in 2010 and see whether those germinates had actually survived and whether there was more germinates as well. So occurrence and simply survival. So the results, the total number of germinates produced at 72 weeks and 14 treatments. That was the other thing. I used 14 different treatments, as you can see there. Just to try to see what did germinate Pimelea spinescens, because we really didn't know, and it was a species that was difficult. So from 2,000 seed, 236 seedlings were produced, which is only about 11%, which doesn't seem that great, but generally they were produced in these ones with gibberellic acid. Things without gibberellic acid really had quite poor results. And what we realized was we could do every single treatment and we get good results, but we could also just use gibberellic acid. So we just used gibberellic acid when we went on and did more germination trials. So this is the pilot study we did in 2008. We also found out another interesting thing when I put them in the 2 incubators. We started one incubator in winter and one incubator in summer. And the summer incubator, as you can see, pretty much nothing happened at all for quite a long time. And then we finally had 12 weeks of summer and 8 weeks of autumn and spring. And when we started to go into cooler conditions, we definitely stimulated germination for the first time. We got these really amazing peaks during autumn and into winter, and then it tapered off as we got into warmer conditions. So the winter incubator did the best, but the summer into autumn also did quite well as well. And interestingly, I sort of left it after that germination peak, and we decided to leave it go through once more into another cool period from the summer back into autumn, and it happened again. So these were exactly the same dishes with exactly the same seed. So I hadn't added any seed. All I was simply doing was adding some water to make sure that they were moist. We got another germination peak that was as high as the first. So it just goes to show that the seed, even in optimal conditions, doesn't necessarily germinate. It might hedge its bets and germinate next time. It's going to be good. So the seed is very viable for long periods, and it will germinate when it's ready. Thank you very much. So there's a seed dormancy, and I found out what the seed dormancy was, which I'm not sure if I've written it here, but I think I've read it somewhere. It's a psychological one. Basically, gibberellic acid will put it in cool conditions. Then I also looked at germinates produced at the sites in 2009, 2010 per female plant, and there were greater numbers with less time since a biomass reduction event. So there were high numbers of germinates at sites that had a 0.5 to 1 year time since the biomass reduction event. And as you went on, there was less and less germinates found in the field in time since the biomass reduction event. So definitely getting rid of that biomass allows a germinate to actually be at a site. And this is per female plants, because they were actually more found next to female plants. The germinations of survival, so from 2009 to 2010, more germinates survived with a higher site condition soil, which is the, it's called, what's the word for habitat hectare score. Basically, I use that. So more bare soil and less weed cover and less organic litter allowed more germinates to survive over time. So that's probably not surprising, but it definitely is interesting that they like these open spaces. They need sun, they need water, and they need space to actually grow and make it. I also was lucky enough to go back in 2008 and revisit all of my PhD sites and quadrats and capture another snapshot of those populations. 2 sites had no mature plants die in the last 8 years, which is interesting. 11 sites had new plants, ranging from only 1 plant to 31 plants within 3 sites. And 3 sites had no new plants found within the quadrats at all. So that was interesting to see about survival and what was happening over time. In the last 8 years, across 14 sites, 2 sites were not included because they were damaged. So we lost 2 sites, definitely lost plants because of accidents, things going wrong, farmers doing something, and a drain was re-drained, and basically a whole lot of plants were taken away. I don't know if I've gone through less. So what I looked at there is what happens is there was an average loss of plants of 12.2 plants lost and a growth of 10.8 plants per year when I average it across. So collectively, we're losing about 1.4 plants a year across the 14 sites. And you have to remember these sites were in Melbourne and also up in the other areas and also across in the west. So there was across a whole different area. So it's a bit hard to say if it's really reflecting what's actually going on. So 6 sites had a population growth rate which was positive, and then another 6 sites had negative, and then you've got these ones in the middle that actually pretty much doing not much at all. They're status quo, they're staying the same. So that was interesting to find. The thing is, one plant, only 4 sites had 2010 germinates survive from the time when I originally went there. So that was interesting, and 3 sites had no germinates at all. And the sites that had new germinates had been burnt in the last 2 years, and 2 of those sites had also had 2010 germinates survive in 2018. So these sites are still being actively managed to reduce biomass via burning, and so they often council fire breaks. And so this is definitely helping those plants and those populations actually keep turning over and keep being self-sustaining and actually sort of timely growing slowly, I suppose is the word I'm looking for. Management implications: seed germinations occurs during wetter conditions. Female plant density positively influences germinates. Germinates are increased for shorter periods since biomass reduction, and the survival increases. So you want more bare soil, less weed cover, less organic matter, which is normal when you're burning a lot, and reduce their biomass frequently, which is something that every single grassland researcher has found. John Morgan found it for rudidosis. It's nothing we don't really know, interestingly. In 2013 to 2018, I was able to do some research at one site, looking at flower. We looked at flower and seed production across 2 areas of one site, and we had treatments of the burn, mow, and control just to see what did influence things. We were able to get burns in autumn 2013 and spring 2016, so it was about a 3.5 year interval in that time period. And I looked, 85 plants were identified across the site from a population of 240 of these plants, and annual seed collection was taken from these sites over time. And we measured that. We found no significant difference between the sites or years, which is interesting. The sites probably not so, but the years were interesting. But after 2 treatments, timing, burning was the only thing that was significantly affected, and it affected flowering and seed production. This is the annual seed production from those sites over time, and you can see that following a burn, we had a massive spike in seed production across exactly the same plants. So I think in 2012, I got 400 seed from 22 plants. In 2013, I got 758 seed from 19 individuals. 2014, 6,000 seeds from 59 individuals, so more plants were producing seeds. 2015, it dropped back down to nearly 3,000 seeds from 50 individuals. 2016, 3,011 was collected from 52 individuals again. And then in 2017, following the burn, we had nearly 11,000 seed was collected from 58 individuals. So essentially, over some 2014 to 2017, the same sort of plants were producing. Maybe some of them weren't, but they certainly produced more seed. So biomass was also studied in this area and found that a greater percentage of bare soil over winter allowed the plants to produce more seed, but further research is required to see if this trend persisted. Another thing I got to do was see if I could establish plants. So I was able to collect, I've been collecting seed for many, many years for my PhD, and I also collected from that particular site over the 8 years that I was doing that research. And we grew plants from them because I could grow them, because I had germination. I knew how to germinate them, and we put them back into 2 different sites. One was a grassland site and one was a flower site sown with forbs, because I thought that would be a really great experiment to see if they did better. So what I did was, and I also tried to do burn, mow and control in those plants that I put back in. So we had randomly selected areas where we had 3 of each at each site. And basically, I'll show you there. This is the flower site that was Greening Australia did this for me. I employed them to flower it. It had been a flower field prior to that. We didn't destroy anything to do this. It was already being ploughed for crops. They ploughed it and then they put grassland seeds in September 2012. And then it took a bit of time, took about a year or so before we got germinates coming up. But we had murnongs, we had podolepis at the site, we had lots of lignum, everything, Tyto spatulatus came up. It just looked amazing, the site when it got going. All the bulbines took over, and then we planted plants, and they did predominantly, this is the site over time. It's quite a beautiful site, and they definitely weeded it for us as well. So kept it open, kept the grasses out and kept it open for us. Maybe a few more photos. And these are the results of those sites. Essentially, the experiment showed no difference between the treatments and the different mow, burn and control within the plots, but definitely the grassland site definitely is not producing or not allowing more plants to live compared to the ploughed wildflower sites. We have 45 plants out of 256 alive in the grassland, so only 22% survived, and 114 plants survived out of 260 in the plough or the wildflower area at the end of 2018. And plants that were planted the earlier they could be in sort of winter did much better than the plants that were planted sort of late winter in June, July. So the longer they were in the ground prior to summer and spring, definitely was an advantage. And also, this is 2015 planting. That's also when I planted it. That's what they look like, the little plants when they went in. They're very small, and that's what that plot looked like in May 2018. So they've done really well, and they all survived. Every single plant in that plot survived, and it was quite happy in that flower plot, which never happened at the grassland site. Another interesting thing that happened in 2016, I think, no, 2018, we had our very first germinate come up all by itself, which we never planted. So that was the whole idea, that we wanted to put plants in and we hoped that they would do their thing on their own eventually. And sure enough, we did get one germinate and it's alive again today, but I haven't been out to that site for a little while now and I'm not sure what's going on, and it certainly isn't being kept open anymore. But during the research, it was, and certainly the plants in the flower plots were doing quite well. So management implications again, greater bed soil promotes flowering, allows greater number of seed per stem to be produced by a plant. Burning a section. The other thing I found, which I didn't quite articulate before, is those sites where the burn, mow and control were right next to each other, is what we found after the burns that went through is the mow and the control plants produced more seed as well. So that burn that was right next door had affected those plants' seed and flower production as well. So it doesn't matter that they weren't burnt. I think that they still produced more seeds. So that was sort of interesting. It wasn't necessarily, we think, the mow or the control obviously had nothing happened to it, but still the control plants produced more seed in the year after the burn had happened. So that was interesting too. Less grass and a greater percentage of bare soil significantly improves establishment potential of transplanted seedlings. So keep your sites open for these plants, weed them, but keep them open, and increase seed. More seed improves the chances of germination. That's pretty much a given. So if you put more seed down, you're likely to get more plants. So the future of trying to augment populations is seed orchards, possibly. So this is something I was lucky enough to get to do as well in 2014. I was given the opportunity to plan a seed and implement a seed orchard specifically for Pimelea senescens. So I had collected seed over time during my PhD. And so I had quite a bit of seed, and I'd also been collecting seeds from the other places and also we'd collected seed from different places around Melbourne. I decided to go, I talked to a lot of people about it. I talked to the Arboretum at Euroa. I talked to people in South Australia who'd been doing seed orchards for native plants, and they basically suggested to put the irrigation under the weed mat, not above it, because you waste a lot of water. So we did that. Unfortunately, the person that flowered my plots was a person that was used to putting in orchards, as in fruit trees. So that's why they're enormous. They were like 2 meters wide, and I would never have made them quite that big. I sort of made them a lot smaller. He didn't quite understand how small the plant was, but you know, you can't be everywhere and you can't do everything. So that's why they're big. And I sort of selected areas that would be where each population would be in a spot, and move them down. And in 2014, I grew a whole lot of plants in the incubator, and I was able to start planting plants into there. That's me and the team in there putting the little tiny baby plants in the seed under the weed mat, and they also had their irrigation system. So we had a system that we could set off when we were there and also set off when we weren't there, just to make sure that the plants actually did some watering. We also had to protect them. We found it was very open at this particular site, so they got quite wind affected. There was no other plants around them, so I found I needed to put some shade cloth around my little cages. Also, we found, unfortunately, that mice loved the site. We were able to keep everything out of the site. We have a great fence around it, no roos, they have stock on the site. They weren't able to get in, but unfortunately, little mice seem to do a lot of damage. You'd be surprised. And that's what they look like when they were all in 2014, 2015. Little plants are all singing away there. They're all growing and doing very happy. I think definitely the year for these plants made a difference too, because we had less plants survive in other years, unfortunately, when I tried to add and augment. And the results in 2018 were that we had about 143 plants alive at this site from, I think it was 17 different populations across the state, and with up to 16 plants from each site. And the aim was for 30. We will keep going to adding and adding to this. And the site itself is huge. I think there was 10 rows, and we weed matted 3 of them, and we planted two and a bit, I think, or 1 and a bit. And it has huge potential, and essentially it's in the Western Grassland Reserve, so I assumed it would be a place that we would use in the future to do augment and grow grassland plants, which unfortunately I haven't been able to get there for a long time. That was the first open day. I got the Pimelea recovery team to come out and people to help me, and people from DELP and Wyndham councillors that came out. It's in their area, and we put seed bags on because it was very quite labour intensive getting little bags on the plants. I collected seed over 3 years from this site, and I still have some of these seeds today that we can use for projects. That was the idea, was collect seed and use it for projects at different places to augment their populations. And that was putting seed bags on again and off. And then 2019, we're back out again. There's DELP, there's Karen. Karen's here today. She came out and helped. And Karen again, and we collected seed bags. The future is probably detecting. We've got, I actually gave a plant to, I think the same Jackson. Jackson had this dog, Kitty, and she's trained up to be a Pimelea dog. So she goes out and sniffs plants out, which is a new thing that they're doing with weeds and insects and all sorts of things that are difficult to find in the field. So that's a great idea and certainly working. A seed orchard is a great idea if you can do it on a small scale, or also help other contractors to do it. That's something I've been working with in one of the regions, was helping a lady who had a native orchard, nursery, and I helped her with gibberellic acid and talking, and she would talk me, and we definitely got it, and she definitely started germinating plants on her own. So that was really great to do. 2017, that was an amazing year for flowering at the seed orchard. And you have to remember, with a seed orchard, you have male and female plants. So not all those plants are producing seed, but you need your males there as well. And also, we don't know what the genetics are a little bit too, because they could be from Ararat, or they could be from Christie's Road, but we made sure they were all from the Western Plains, because the genetic research, which I seem to have omitted somewhere here, we found that the plants beyond the Great Dividing Range, so the areas around Terrick and those populations up there, are different to the plants from Melbourne across the Western Plains. Sorry that I've omitted that slide. I'll probably somehow have left it out. Signage is another one that we think is really, really important. If you can put some signs up, it might be your last chance of possibly stopping some destruction occurring. I mean, that's a huge sign. That's probably not, hopefully, but it is good to wait to give people some, this is what's here, and please come and have a look at it, because a lot of people won't get out of their cars. This is a great one around Lara. That's where the air show is, and they put those signs there to stop people from parking on the grasslands during the air show, and it's working. And the people who are, there's some businesses right next door, and they're really proud of it, and they don't want people parking there. So they also put flagging tape up around those signs, and from there, so people know why you're not parking there for that sign. So it's good to just let people know, and people are happy to support it. That one's in Wyndham for an area of the road that gets dumped on continually. So they put up a sign to possibly give people a bit of a hoy, please don't do it. It's not the best thing to do, and possibly tell them no. That's a great sign. I do like it. It's quite bright and poppy, and certainly it tells people what's going on. And this is what we recently were able to do with Trust for Nature. With DOB applied for some funding, and they replaced a lot of signs around the state in the Bendigo region predominantly. And I know we've actually helped give these signs to some of the other councils and some of the other DOB areas to use them, because they actually are clearly saying no mowing, don't dig, we don't want cattle on there, and don't weed spray because it's endangered grasslands. And we've also got a nice picture on it to show them there's something pretty there to look at. And that's replacing signs that actually had nothing. They'd faded to nothing. Just a signpost with nothing on them anymore. So that's good, that was better. And this is another really good one, I think, that's actually at Wycliffe, just near the cemetery at Wycliffe, just giving some education to people that there's some amazing areas here and what's here and what we're doing. And just alerting people, I suppose, what they could see and why is it important. I think it's great for education for everyone to understand what's there and why. And certainly Shires and people doing it. It's Glenelg Hopkins doing that one. The other thing I've been able to do is augmenting secure sites, I think is another really important thing we should be doing. Secure sites that have small populations that could be augmented. We need to be moving the genetics around a little bit. The plants around Melbourne's population has probably the most global warming adapted, because it's in 3 rain shadows. We've got the Otways, we've got the Brisbane Ranges, and we've got the Yuyangs, essentially. They are all shielding sort of the Keeler plains for water. So those plants specifically are a lot more water adapted and a lot more hardy, and probably we need to be getting those genetics out into some of the Western plains, out in the all the Rokewood areas and those areas, just in case, because if they start suddenly drying out, they're probably not going to be as adapted to the really dry conditions that are coming, possibly. This is Arcade Way, and I worked with Millie in the council, Moonee Valley Council. We only had 2 or 3 little plants here, and we planted 10 of them, and we doubled the population. We still got a bit more work to do there, of course. We want to keep going at that and putting more plants in it. That site's getting actively burned. There is some remnant Blue Devils, and I think there's some goodenia there. It's quite a good site, and it's certainly within Metro Melbourne, so it's going to always be there. So I think it'd be amazing to augment it and make it better for everybody. That's putting some protection on it, so it's not accidentally mowed or laid on. Oh, that was Pioneer Park. Oh, I should have back. That was actually Pioneer Park, which had 4 natural plants when I first went there originally, and we were able to translocate 54 in 2019. So they've been augmented. Not all of those plants are survived. Oh, yeah, 17 are alive today. And also Megan and I were able to put in, I think we ended up putting about 56 plants in that were babies from different areas that we grew, and I think possibly about 18 or 20 of them have survived as well. So now that population is quite well augmented from lots of different genetics, and also much more secure, and probably the population itself has a bit more robustness about it for the future. And Skipton Common is another one that the Ballarat Environmental Group asked for funding through Trust for Nature, and they wanted to they didn't realise they had Pimeleas there at all. This area had been traditionally sort of grazed. There was lots of fences around. They removed all the fences, took the grazing off, talked with the cultural people of the area, the Aboriginal groups, and they have had a burn there now, which is amazing. And they also sequentially tried to count, and they found that they thought they had about probably 300 or 400 plants, but inevitably they had 1,367, and we also augmented their population as well. So they had a very active school. The school group goes up there and counts the plants every year, and actually takes an accounting of what plants are in flower and what is there. They also have a land care group and lots of locals. So it was really cool putting them back. And we finally got out there in 2021 to plant plants, because we've grown them and it's been a while, and planted 49 plants. I'm not sure how many exactly have survived, but they definitely have different genetics to try and augment that site. So that was a really great thing. And I think that's the future of it. I'm really happy to try and do that with people. Definitely my role at Trust for Nature has changed a little bit, in that I'll actually be able to go out and help people with their populations, give advice, help with seed collection possibly, or help coordinate and help you to do it for yourself, and also growing plants possibly, and putting them back in the ground. So please reach out to me. That's sort of part of my role, and I definitely want to do it in the future, because we think augmenting and learning, if we teach people, they will tell other people and teach other people how to do it as well. And here's some pretty pictures of Pimelea spinescens, essentially. This is a germinated seed, germinated plant with a little seed cap on. That's what they do. They have these really cute little green cotyledons and their little seed caps hanging in there. It's so cute. And there we go. Then we pop off and we've got the two little cotyledons, which are quite they're quite succulent when they come out. The little seed cap's still attached. And again, it's a little seed cap. And you can see how they bolt their roots. They have very, very small amount on top. That would be the soil surface. And their root is probably easily 2 or 3 centimetres long. So when they do germinate, it's in winter when the soil's soft, and they're trying to get the roots down very, very quickly, as fast as they can, to that lower profile that's a lot more moist, and see if they can survive summer. There's a really pretty picture of a male with this little pollen on view. And this one's an interesting one. You can see that there's male flowers here, but there's seed. So that's probably a hermaphrodite. It's producing seed, but it's also producing pollen. And we did find that the hermaphrodites weren't necessarily producing as great a quality seed as the true females, essentially. But they're still valuable within the population. And here's a few pretty pictures of grasslands, which I just love taking photos of grasslands, as you can imagine. That's actually, where's that? Springfield or Springthorpe, I think it is, out the back of Rokewood, one of the roadsides there that is mowed and burnt quite regularly. That's just near Rokewood Cemetery. That's Lucas and Albicans and Flower, looking really beautiful. That's Whitcliffe Cemetery, which the sign was at before. And that's all the little purple bits you can see there are orchids that weren't even open because it was a cloudy day. And it still looks magnificent, that particular photo. Podolepis, I think that is actually Calder, near Calder Raceway there. There's a whole huge bunch of Podolepis right to the side of the road at Organ Pipes there when you drive along. It's not quite as big as that now, I don't think, but that's where it is. And that'll be a grassland somewhere in the Rokewood area. I'm not quite sure where, with all your leucochrysums. And that's the seed orchard. Sorry, that's the plough plot where I planted my plants. They're the plots, and that'll be Podolepis with all its seed heads happening there, and some lichen in the front. There we go. Hopefully that's all right. Hopefully I haven't gone too over time, or hopefully I've gone what you thought I should.
That was brilliant, Deb. Thank you so much for that. It's your a wealth of knowledge. I had no idea that you had done so much extensive research on that. Does anyone have any questions?
Hi, Debbie. My name's Arthur. I'm a junior environmental consultant.
Yep.
I live in Little River, so I'm familiar with a few of the sites that you've been talking about. I was just wondering, from a management perspective, particularly for degraded grasslands, has there been any research into herbicide application, broadacre spraying and its effects, and if there's any kind of sweet spot with low dose glyphosate or anything like that and its effects on Pimeleas?
Pimelea doesn't like, it won't tolerate glyphosate. But in saying that, I know that I'm pretty sure that Pioneer Park was glyphosated, and there was a few plants that survived, but it definitely doesn't like being weed killed. And definitely we lost so much of our forbs there. All the grasses survived, but we've got a really low forb load at Pioneer Park because they did do pretty much a glyphosating of the whole place, which was a bit of a disaster. So I think mature plants would cope with it, but you wouldn't want to, you definitely lose young ones and babies possibly, so you don't really want to be doing it, that's for sure.
Yep, okay.
Other questions, anyone?
Oh, there's lots in the chat, I can see.
Oh, we've been gas bagging away. We have. How long are the seeds viable for? There's a good question.
Yes, from the my PhD stuff that I did, I definitely germinated seed up to 6 years after I'd collected them. They definitely aren't as good, I think, over time, possibly. Not as many seeds are germinating. Certainly Megan will probably vouch for that too, that we're using my PhD collected seeds, and some of the ones for the older ones really aren't that great anymore. But interestingly, with seed, which I didn't really go into a little bit, was because I collected seed from plants at the same sites at least 2 years running. And then I actually did it in 2018 as well. And with the other research that I showed you, where I collected seed from like, I think it was six years at the Burnside site over those plots, I had plants that produced lots of seed and lots of good seed. Like plant 105, I know exactly which plant it was. Every time I did a little dish up with all the seed, I put say 20, 25 seed in a dish, I can guarantee 3/4 of those seed would germinate, which was unusual. Most dishes you've got maybe 2 to 5, maybe 6, that was good. But with particular plants, they were like super females, we used to call them a little bit. They were just really good females who produced a lot of really amazing seed that germinated well. But that was a bit hard sometimes, in that you don't want lots of plants from 105. You want to mix them up and you want to spread them out. Do you know what I mean?
Yeah.
All the same germinate, same genetics. So we needed to be a little bit careful. And it was the same at other sites. Fight had one plant that produced amazing seed, that every time I got seed off that, we had really good production of seed in the dish with exactly the same treatment that any other plant got as well. So it wasn't like I was doing anything special.
Follow-up question to that is, did you do anything like cooling the seed before planting it? Do you have to cool the seed before planting it, like to mimic winter conditions perhaps? Or
No, we germinated them in winter conditions in the incubator. That's the other thing. I didn't really go into the incubator as well, but we started an incubator. We had average conditions for winter. I think it was 13 degrees during the day and 5 degrees at night. And we kept it steady like that. And it was literally lights on, lights off, 12 hours. Or actually, I think I changed it to 10 hours of light during winter, and less light during sort of the winter. And interestingly, in the incubator, if you left the plants in cold conditions, they will just keep germinating. That was what was even more bizarre, is I could have those dishes in winter conditions for 4 or 5 months, and they would just keep germinating. They don't seem to care. They don't seem to want to come out, and they will just keep doing it. The other thing was interesting, when we did the roots, we collected a whole lot of plants, and they were all in bags, and they were with the roots, and I had to do all the studies of them. And we put them in the freezer at the university. And when I pulled some of them out after, a few months later, or a month or whatever, they were still growing. The little ones were still alive. They were happily just producing new stems, and even though they were sitting in pitch black in a 5 degree, -5, 2 degree fridge, it was essentially at the university. So they love cold conditions, essentially, I think.
You mentioned at one point that you did a burn, and the control area next to the burn had a higher rate of seed and so on as a consequence. Yeah. What do you think was causing that? Is that carbon?
It could be the burn, but also I think it was the fact possibly that pollinators were attracted to the plants nearby.
Right.
So they were there with the plants that have produced amazing flowers afterwards, and were getting all pollinated, and then they were also just incidentally visiting the ones next door as well, probably. I think that's what it possibly was. That's a bit of a guess on it. But it was interesting that it's sort of the burn in the area had raised all their seed production, not just the burn plants. I mean, that's what was interesting, is that the mow and the control plants that produce seed also produced more seed that year as well, even though they weren't given the treatment as such. So it is interesting that I think probably the pollinators, but also just the smoke, maybe it does affect the plant. I don't know. It's hard to know.
Do you have evidence that mowing or fire is better than mowing, or mowing is better than fire?
That's what I'm saying. The only significant difference was burning. Mowing essentially actually wasn't any different to control.
Okay.
When you did the stats, it was a difference, but it wasn't significant. When you do research and you put the numbers in and to crunch it all, it was interesting that it actually came out to be no different. So I don't think it's wrong. I think mowing's better than nothing, absolutely. But I don't think it's burning is what we actually should be. If we want to do good grassland management, we should be burning.
What time of year were the burns undertaken?
Well, I did say that one was in spring and one was spring, one was autumn. They were different. And really what we advocate, we have got a recommendations from the recovery team. And really what we know is it's so hard to get a burn done that any burn's better than no burn at all, no matter when you do it. And I mean, if you can get a burn done, even if it is in winter, it's better than nothing, but it's unusual. Like, it's usually harder to do it. So I definitely think early summer, late spring is actually the best time for Pimelea if you just want to look after Pimelea, because they are they've had their seed production, they've had their flowering, they're producing seeds, and then as we head into the slightly warmer time, they'll drop their seed. And I think the plant itself, that's it, it's done its cycle, and actually all the biomass it's got on board, I think it's happier if it's not got it all on board for summer. So if it gets a burn event go through, then the seeds on the ground will just go to the ground, it won't have a problem, and the plant loses a lot of its biomass and doesn't have to look after it over summer. And then it starts growing back in autumn the next year and produces seed again. So that's a good, definitely in along the Western Plains, in the Rokewood areas and Shelford, all those areas, that's when they burnt. They actually burnt for 50 years running, just prior to after their harvest, which is the end of spring, and it was also we call burn fire breaks for their properties for summer, and that's what they did. And I think it actually helped Pimelea and species that don't mind that, but it definitely would have affected other species that flower during that time.
Fantastic. Any other questions, anyone? No, well, perhaps we should wrap this up. It's we'll move on to a Grassy Plains Network meeting in a second, but perhaps if we could all just thank our magnificent expert Debbie for her work, that would be brilliant, or just wave or whatever, or smile into your cameras.
Andrew, sorry, I just had one quick question. The rice flowers that are in Sharp's Court in hoppers that have been put in there. I haven't been down there to have a look, but I think I'll go down and have a check them out, see how they're going.
Sorry, I missed that because I actually put my headphones on and they're working because I used to think.
Sorry, Debbie, just quickly, you can give us an update on the spiny rice flowers that are down at Sharp Court in Hoppers Crossing with Nature West.
I don't really know much about them myself, to be honest.
I can.
There we go.
Megan.
So I just today got an email from Nature West. So I think we put 20 plants in and we've got 3 that are actually surviving, is the count. So, you know, it could be a lot better, but there's 3 there, which there were none there before. So fingers crossed they survive.